In this video, we'll be looking at a few practice problems where we apply the skills that we have learned in these past few videos. Remember back earlier to the centrifugation tubes, where we discussed that the 15, or the heavy mark, was lower, the 14 mark was higher. So what does a dispersive replication band look like? As we discussed before, because dispersive replication jumbles up all the DNA fragments and then puts them back together, our line is going to be somewhere smack dab in the middle of the two. But it's also moving upwards towards the 14 because after subsequent replications, the percentage of 15 is going to go down in the strands and the percentage of 14 is going to go up. So again, we have a strand somewhere in between and it's moving upwards. So are we going to have anything at that 15 in line? No, we're not. So what's the answer to this question? No, we're not going to have any band at the 15 line. So let's suppose that the S. pneumoniae from Avery, McLeod, and McCarthy's experiment actually contained a protein genome. Which mixture of enzymes below will we expect to see the results of a protein genome with? You can pause here, think through these, and then we'll run through it. So let's look at this first one. If we had RNase, protease, and DNase, what would happen? If we had all of these, it wouldn't matter whether we had an RNA, protein, or DNA genome. We would lyse all of these, and we would have invalid results, and thus the mouse would survive. If we had protease and RNase, we would destroy the protein, and again, if we had a protein genome, if we destroy the protein, we would have no transformation, and thus the mouse would survive. If we had RNase and protease, again, we're destroying our transforming substance, we're destroying the genome, so there's no transformation from that non-virulent to the virulent strand, and thus the mouse survives. Finally, with this last example, answer choice D, we have RNase and DNase. So RNase and DNase are going to destroy any cellular DNA or RNA, but they aren't going to destroy the protein, and the protein is our transforming substance, and thus this is going to kill the mouse. And so it's this combination of enzymes that would have proven that this protein genome was the case in Avery, McLeod, and McCarthy's experiment.
Now, let's apply what we learned in the Hershey-Chase experiment. In an experiment on bacteriophage protein expression, the small viruses are labeled with 35S. A culture of bacterial cells and phages is lysed and centrifuged. Where can we expect to find these proteins if fractionalization matched that of the Hershey-Chase experiment? So now let's look at these answer choices. Could it be the nucleus, the supernatant fluid, the nucleic acid pellet, or impossible to determine? Let's first think about why would we want to use sulfur to label proteins. Take a second to think about that. Let's take a look at these amino acids. Amino acids are the monomeric building blocks of proteins. And these amino acids, cysteine and methionine, both contain sulfur in their side chains, or R groups. So, now that we know why we were able to label proteins with sulfur, let's look back at the question. Now, which one of these answer choices is going to be indisputably wrong? Let's look at this. Let's think about, does a bacterial cell have a nucleus? No, it doesn't, right? So we can eliminate this answer choice right off the bat. Now, let's think back. The question says that this matches the conditions of the Hershey-Chase experiment. Now, even though this isn't exactly looking at what the Hershey-Chase experiment was looking at, we know that from the Hershey-Chase experiment, in our fractionalization, we had a pellet that had the DNA in it, and we also had supernatant fluid where the protein content was found. So we know it's not impossible to determine, and based off of this diagram right here, we can conclude that the protein content would be in the supernatant fluid.
I hope you found this video really helpful. The concepts and information presented in these videos will be true no matter what genetics class you are taking. However, the concepts presented in this video are referencing material currently covered in Baylor University's coursework. Remember, if you are currently enrolled as a Baylor student, we offer free tutoring services. Our tutoring center is located on the first floor of the Sid Richardson building.
You will find all the details you need to know about these services on our website, www.baylor.edu/tutoring. You may schedule a free 30-minute one-on-one tutoring session online through Navigate, or just drop in during our open business hours. For more information about our current services, please visit our website. Thank you.